Dear audience, I am very, very happy to welcome you all here. I know that more people are going to come here. I'm quite a big stickler for the timing, so we'll start on time because we have 5 amazing speakers today. And I would say this session will be a little bit different from the morning sessions where we were listening a lot about the genomics in the precision medicine. So I am very happy to show you that there is other side of the precision medicine as well. So that is why our section is called Functional Precision Medicine. And to make the point across, I have 5 amazing speakers, and I am very happy and honored to invite our first speaker, Professor Anthony Letai. He's a professor of medicine in Dana-Farber Cancer Institute, Harvard Medical School, and associate member of Broad Institute in the USA. Please welcome.
Good afternoon. It's a pleasure to be here in Latvia, a country I've never visited before. In fact, I've never visited any of the Baltic states. So today I'm going to talk to you about functional precision medicine. First, I'm going to talk a little bit about what I mean when I say precision medicine, and then, of course, what I mean by functional precision medicine, and why I think we need it. And then I'll also get into a little bit of I'll talk about the field in general. Then I'll speak a little more specifically about some work that we've done in our laboratory using some of the concepts and tools that we have. But I'm very happily followed by a couple of people who a long time ago I used to supervise more directly. Both Patrick and Joan will also be talking about this. Here's some disclosures that I have, a few scientific advisory boards and some funding I've had from pharmaceutical firms that you see there. Okay, so I think we need to remember, I mean, this is my definition, and I think, if you agree with me, I think we too often forget about the fact that you're really not doing precision medicine unless at the end of your study you are matching a patient with a drug. I think there's something attractive, trendy a little bit about the use of the phrase precision medicine, but I think all too often it's like sometimes a very good scientific study, maybe, terabytes of information, omics and UMAPs and all these sorts of things. But at the end of the day, you've provided a whole lot of information, and I don't see always that there's an actual drug chosen for a patient that actually helps that patient. And if you're not choosing a drug for a patient, I don't think you're doing precision medicine. You may be doing very creditable, very useful research, but precision medicine has its own words, and I think we need to respect that a little bit. What's functional precision medicine? Well, a lot of what we heard, including genomics, are what you might call static or sort of initial condition assays, which is that you take a snapshot of some state of the tumor, maybe you look at the DNA or maybe the RNA or the protein or something like that, and you take a snapshot and try to predict the future using that snapshot. Functional precision medicine, we think that if you want to find out if a drug works, the simple thing you do is you take the drug, you put it on the cells you're interested in, and then you measure something that tells you whether that drug is going to work or not. It seems very commonsensical, doesn't it? That if you want to know if this drug is going to work on this cell, put this drug on this cell and see what happens. And it is commonsensical. In fact, even nowadays, today in 2023, this is the gold standard for choosing antibiotics in microbiology for people with a bacterial infection. This is just an example of what it might look like. Here we have a lawn of bacteria that you can imagine was cultured from a patient, maybe with a urinary tract infection. What's the best drug to give this patient? Well, you simply expose the bacteria to all the drugs and see the ones that work. So you might choose a drug here that's killing a lot of bacteria, but you would avoid a drug there that isn't killing bacteria. Very simple, it's sort of a low data readout, but highly, highly actionable. And that's the story about functional precision medicine. It's relatively small data sets, but the end of the output, but it's very, very actionable because it's always about matching a drug to a patient. So if this is so commonsensical, why don't we use this? Why isn't this in general use? Because it's not a standard test nowadays. So I'll tell you, back in the 1990s or so, people were doing these sorts of things. They called them ex vivo chemosensitivity assays. And a lot of them were testing whether or not a particular sample was sensitive to platinum-containing chemotherapy agents. And these so-called ex vivo chemosensitivity assays, if you look back at them, do show some correlations between the in vivo and the ex vivo experiments that were performed. But the correlations weren't superb, and also there really weren't any prospective clinical trials performed to demonstrate that there was sort of clinical benefit from people receiving these assays. So there were a lot of claims made by these assays, but I think there was definitely a lot lacking in terms of evidence. There were also technical limitations. We knew much less about culturing cells back then, and a lot of the cultures were overgrown by like fibroblasts. There are very few drugs to choose among, so it wasn't like you were choosing from a whole palette of drugs what would work best. It sort of, did platinum work or not, for instance. So you can see starting, I think, even earlier than 2004, I have a couple examples here, where the American Society of Clinical Oncology, ASCO, put out a couple position papers saying, and I think I basically agree with them, that these chemosensitivity assays were not standard of care, and there needed to be more evidence developed before they became standards, okay? What else happened that sort of pushed these very common sense chemosensitivity assays of the way? Genomics happened. And I say it conquered the 21st century because it was really at the dawn of the 21st century that the people really realized the potential of genomics, not just in cancer, but throughout medicine. And it started with the actual completion of the first human genome by both the NIH and Craig Venter. As you can see from this headline right here, that's June 27th, year 2000. And then in cancer specifically, something really, really important happened. And that is we realized that imatinib, a kinase inhibitor of the Abl kinase, was incredibly successful in treating this difficult disease, chronic myelogenous leukemia, that we could only treat with bone marrow transplant really successfully up until that point. And it turned out, we now know, you can give patients with chronic myelogenous leukemia, CML, imatinib, and they essentially live normal lives. They have to take the imatinib for the rest of their lives, but it's an absolute game changer. It radically changed the treatment of CML, saves thousands of lives every year in the United States alone, let alone worldwide. So that was really big. And what people understood this was a case where you took a genetic alteration, which was a translocation event between the BCR and the Abl genes, and activated the Abl kinase, and then you targeted the Abl kinase, and it occurred to people, well, this is it. This is how cancer is finished. This is how we're going to do it from now on. And you saw a lot of this is from the NIH, but you saw a lot of figures like this from cancer centers, including my own at Dana-Farber, where you could see that the solution is plain as the nose on your face. You're going to identify mutations like that. That's what that is, or that is. Then you're going to mix that. You're going to match that purple mutation with a purple pill, or that green mutation with a green pill, the orange one with an orange pill, and we were going to be done. We're just going to do for all the cancers what we did for imatinib, right? And in fact, early on, there were successes, okay? Some significant successes, probably notably early on, was for EGFR, targeting EGFR mutations in lung cancer, and then c-KIT and GIST, and you can see the whole list up there. And in each of those cases, there was very real patient benefit, and I don't want to say otherwise, okay? However, it wasn't quite like imatinib, was it? What happened in almost all these cases, not all of them, but almost all of them, you got much more transient benefit. A patient was treated with a single-agent kinase inhibitor. There was very often a response, and then inevitably a relapse sometime, maybe a few months, maybe a year or two later. So there's real benefit in these patients with advanced cancers, but it wasn't like imatinib. So this didn't look like it was totally going to be the solution. So people started to study, maybe we can do big gene panels and maybe choose among a whole lot of drugs, and maybe that will help. And there were a number of trials, and I'll go over in a little bit of detail here, the NCI MATCH trial. And my purpose here is not unduly to criticize genomic precision medicine, because I want to emphasize it really does help people. But I think all too often we're seeing what I call the numerator, not the denominator. We see the success stories. We see the pictures of the people whose tumors shrink away. But we don't always see the denominator, all the patients whose genomics were studied to identify that one rarefied patient. So here's an example from the NCI MATCH trial. This was a trial that was started several years ago. run by the NCI, but it was throughout the United States, where thousands of patients eventually had their tumors sequenced, their advanced tumor sequenced, and then they were supposed to be allocated to the arms, one of several arms on the trial. It started out with 10 arms, and it wound up with, I think, something like 36 different arms. So early on, I sort of did a brief review about how it was doing, and what's really important to look at is like how many people are benefiting. And I tried to draw, this is early on, and you can see that early on there were about 700 or so samples were identified and they were sequenced. And then you can see some of those, they actually were able to complete the tumor testing. That's the internal one. Then of those, only a few of them actually had a mutation that allowed them to be assigned to an arm. Even fewer got actually onto a treatment arm, and that left us with only 16 patients out of the 700 some who were studied who actually made it to a treatment arm. And I have to say, the reporting coming from the NCI match trial didn't always emphasize this fact. It was sometimes hard to see that this is what was going on, and this didn't even cover what response rates were. So what were the response rates? Well, we have an update, a really important update, and the citation is there. It's recently in Nature Medicine, of the NCI match trial, where they have completed the over 6,000 patients enrolled and almost 6,000 genomes sort of studied with their genome panel. 38 arms, not 36. 8 of them closed because they just couldn't fill them up. So they reported the results of 27 of the arms, okay? 765 of the patients were assigned. So already we can see we're pushing just a little more than 10% of the patients even made it to an arm of this trial. That meant nearly 90% of the patients didn't even have a mutation that they could link to something that could get them to an arm, or something else happened that they weren't able to get there. They reported in this paper with a signal-seeking goal. They claimed this was their main goal, was to signal-seek, and that is to identify arms, and it's a legitimate goal, to identify arms where there was over a 16% response rate. 16% though, I mean, think about it. This is already, I think, somewhat of a lowering of the bar of our high expectations early on for genomic precision medicine, right? But that wasn't really what they identified as the primary objective, which is a little puzzling why this isn't reported a little more clearly. The primary objective is to evaluate the proportion of patients with an objective response. And I challenge you to go to this paper and actually do that mathematics and figure out, well, what is the overall response rate? Well, I did it for you. The total number of responders, people who had an objective response in this trial of over 6,000 patients, was 79 patients had objective responses. So that's, if you treat this as an intention to treat analysis, which I think you should, about 1.3% of the patients who were entered on this genomics trial actually had a response. This is not a real outlier. I think we heard similar figures from some very good talks early this morning, where single-digit percentages of people are actually getting clinical, observable, objective clinical benefit from genomics, which is not to say they're not getting benefit. They definitely are, but we need something else. Here's a couple other trials, if you want to look at these, where they actually looked at how well a broad genomics trial was doing, the Shiva trial, which is somewhat old, and the Moscato trial, which is maybe a little bit newer. Low numbers of patients benefit. Most patients didn't benefit from a purely genomic precision cancer medicine approach. So I want to caution everyone, when we talk about cancer medicine and precision medicine in cancer, there's a tendency to make it synonymous with genomic precision medicine, okay? And while there are definitely life-saving and life-extending consequences in some cases, it's not in most cases. Most patients aren't helped by genomic precision medicine. And the main point I want to make is we can't really expect major progress in precision medicine and cancer if we only use one tool, and that tool being genomics. We've sequenced hundreds of thousands of cancer genomes right now. There aren't new secrets waiting for us to unlock that's going to affect a lot of patients. We know what's going to happen. There's relatively few patients who benefit from this. Sure, there will be more if we have more drugs, and there's different things that we can do. But I think we need to take a much more sober look at a purely genomic approach to cancer precision medicine. What do I suggest? Let's reconsider functional precision medicine. And this is a field that has really sprung back to life in the last 5 to 10 years. And part of the reason is because we have many more drugs to choose from. Instead of just platinum or nothing, we can choose between a wide variety of all these different targeted agents. And here I've listed a whole bunch of different targeted agents, and there have been 88 new drugs approved in the United States, at least, since the year 2000. So a lot more to choose from. We also have a lot more different ways of studying them. We study them. There's some really cool assays that study in situ. That's right, directly inside the patient. You put the drug and then sample later to see if the drug worked, or a set of drugs. There's 2D cultures, 3D cultures, microfluidics, cell mass. PDX models, I think I'm not going to talk much about those. I don't think they're a practical solution. It's too hard and too slow to really grow everybody's tumor up in a human or even fish, in a mouse or fish PDX. There's also a much better variety of measurements. We now have a much far greater capacity than we did 20 years ago of doing single cell measurements, which is very important in heterogeneous mixtures of tumor cells. We can also measure all sorts of different phenotypes, including those that are assisted, artificial intelligence assisted measurements of morphology. Okay, I can't get into all of this. It's a short talk, but if you are interested in this, I encourage you to join us, or at least check out the Society for Functional Precision Medicine, which was founded in 2017, and I have the pleasure of serving as the president of this organization. You can, if you're interested, point your phone right now at that QR code, and you can visit our website, but it's a very easy one, sfpm.io. And check out our monthly seminar series, or consider joining, or drop us an email. We'd be very happy to hear from you, very happy to have you get to know us. I just have sampled here just a few, does it work? Is there evidence for it? Yes, there is. It actually does work. And here's a bunch of different citations. I'm not going to list them all. These papers really speak for themselves. But there's growing evidence that it actually does work, makes patients live longer, and improves their progression-free survival. So another thing you'll notice if you look at these studies is it's a far greater proportion of patients who actually make it to a treatment arm than you see in the large genomic studies. So that it's much more likely that you'll actually find a drug that works for a patient than if you rely on the genome alone to tell you how it works. So with my remaining time, I'm going to talk to you more narrowly about how my laboratory approaches this functional precision medicine. We study the apoptosis field, and what we basically have is an assay that can tell us in a very short period of time whether a drug initiates an apoptotic signal. And we've also shown that if you do identify a drug that's initiating an apoptotic signal, it's a good predictor of actual clinical response. So this is the apoptotic pathway, and the regulation of apoptosis is done at the mitochondrion. When the mitochondrion permeabilizes, you get a commitment to programmed cell death via a lot of events I'm not going into. And this permeabilization event is regulated by the BCL2 family of proteins that has both pro and anti-apoptotic family members. The pro-apoptotic family members actually form the pores, and the anti-apoptotic family members hold them at bay. And the way they hold them at bay is there's an interaction between the anti-apoptotic proteins have a hydrophobic groove, and into that groove binds the so-called BH3 domain that has a hydrophobic face. So it turns out that BH3 domains can be made into BH3 peptides, and these exert a lot of the same pro-apoptotic activity. So they're in some ways the currency of apoptotic signaling, sort of like ATP is a currency for energetics. So we found out a very long time ago is that you could use these in assays by applying them to mitochondria, and you could ask, how much of this BH3 peptide do I need to add to mitochondria in order for the mitochondrion to permeabilize? And that gave you an indication of how close a cell or its mitochondria were to the threshold of apoptosis. So if you needed a lot of this peptide, of these BH3 peptides, to cause a permeabilization event, that meant that it was very far from the threshold of apoptosis. If you only needed a little bit, that meant it was close to the threshold of apoptosis, what we called being primed for apoptosis. Well, if we could map the proximity of a cell to the threshold of apoptosis, we could then answer the question, does this drug move the cell closer to the threshold? And this is what we call dynamic BH3 profiling, what I'll be telling you about. And what we do is we take cells and we treat them with a drug and compare them to a non-drug treated control. And if a drug works, like you see here on the left, then what you see is that the drug moves it closer to the threshold. So when we add our BH3 peptide, we often use one that we call BIM-BH3, it's enough to push it over the edge. And we read out mitochondrial permeabilization by cytochrome C release or various fluorescent dyes. We can discover that the drug actually did something to make it more sensitized to apoptosis that we sort of develop using this BH3 peptide. If the drug has no effect, the cell sort of stays where it started out, and we apply the BH3 peptide, it might move it a little close to the threshold, but not over the threshold, so we don't actually read anything out, and we don't see a signal. These signals develop in just hours, which is important because it means we can study primary patient tissues without needing to sort of set up very extensive long-term cultures that have, I think, a lot of liabilities. This is an example of how we handle mesotheliomas, but all solid and liquid tumors are roughly handled the same way. We make a single-cell suspension, pipette it to a 384-well plate, incubate them with drugs overnight, and then the next day we permeabilize the cells so the BH3 peptides can contact the mitochondria, and then we measure whether cytochrome C is released or not. And when we compare the cytochrome C release in a drug-treated versus a non-drug-treated sample after we apply the BH3 peptide, we get something that we call delta priming, and the higher the delta priming, the more active the drug is, basically. So now I'm just going to run through a bunch of examples for how broadly this worked. You need to make very few assumptions about the drugs that you're using or about the cells that you're testing in order for something like this to work. You're just asking the question, does this drug induce apoptotic signaling compared to an untreated control? So here is acute myelogenous leukemia. This is a set of assays that we did on samples that could be made into PDX models, patient-derived xenografts in mice. And this allowed us to treat the same sample many different ways in vivo. So we could make ex vivo predictions and then test them in vivo. This was work done by Shruti Bhatt, who's now at the National University of Singapore. And you can see whether the drug is birinapant, a so-called SMAC mimetic, epigenetic agent like JQ1, a BCL2 inhibitor like venetoclax, or a kinase inhibitor like quizartinib, very different classes of drugs, but they all signal via apoptosis, that we're able to correlate very well the signal on the x-axis that we determined from our assay, the delta priming, versus the actual shrinkage of the tumor in vivo over 15 days. And this is a receiver operating curve over all the mice that she studied. And the lesson here is in AML, at least, as we show you here, there's a very good correlation between our assay and our ability to predict what the actual in vivo response is. Here's a set of ALL, acute lymphoblastic leukemia models, treated with a very different drug, an HDM2 or MDM2 inhibitor that activates P53. And these are the actual clinical, if you will, in vivo responses. And Joan Montero did this experiment. By the way, it's Joan's birthday, so when he gets up, give him a big hand. Very good correlation between responders and non-responders. And you've seen a couple receiver operating curves that tell you the trade-off between specificity and sensitivity. And we had a perfect receiver operating curve reflecting our ability to perfectly discriminate between those samples that eventually yielded a response to those that didn't in this very different tumor. We don't do just leukemias, though. Here's a few more leukemias. We can predict response. This is actual clinical response, not looking at mouse models. This is actually human beings. This is our prediction of response to venetoclax in AML. This is a complicated regimen in the middle there of lenalidomide plus mitoxantrone, etoposide, and cytarabine, where we did BH3 profiling the same way. Again, very good discrimination between responders and non-responders, actual clinical responders. And this is old work also done by Joan, where he studied our ability to predict response to imatinib in CML, that targeted agent I told you before. Again, excellent predictive value that compares extremely favorably with predictive biomarkers used today in the clinic. This is FLT3 positive AML. Again, in the clinic, we're able to discriminate between the non-responders and the responders very, very efficiently. I want to point out that we have a clinical trial that we hope to start hopefully within the next 12 months, where we're actually going to use dynamic BH3 profiling to assign AML patients to different arms of a study where they'll actually be assigned therapy according to whether or not what the signal is from our dynamic BH3 profiling platform. So I'm going to wrap it up now. What I see the future is, of course, we should continue to use genomic information. We should also use whatever information we have from hard-won clinical experience of the drugs that work in a particular genotype or histology. But we also, I think, it's inevitable. This is how precision medicine is going to advance. Of course, there's going to be logistical hurdles to overcome because you need actual viable tissue to do these assays, but they must be overcome because the information you get is so valuable from these assays. What does this mean for Latvia? I'm in Latvia. Well, I'm very excited to hear that there's a broad sort of countrywide approach being taken towards precision medicine. I think it's essential, and I implore the people involved in this project to include functional precision medicine in your plans. I think you have a real opportunity here as you're building up to build this in from the ground up rather than having to retrofit an existing system of genomic precision medicine that only serves a small minority of your patients. Why not build right into it a very exciting new platform that can also help, I think, a much broader section of patients? So I'll conclude here. Long list of people who helped with these studies, and I can't name them all. I tried to name them as I go through. And as we change over, I'll just give you another shot at this if you didn't get a chance at our website. So thanks for your attention, and I look forward to talking to you a little later.
Thank you very much. So to follow Tony's talk, I invite Patrick Bhola. He's the head of the Laboratory of Functional Precision Medicine at Harvard Medical School. Patrick, it's all yours.
Great, thank you. Thank you. So I'm an instructor in the Letai lab, and I'm also the head of the Laboratory for Functional Precision Medicine at Harvard Medical School and Dana-Farber. So there's two main points that I want to make in this talk. First is that we've developed a rapid and functional cancer biomarker that can be used on small diagnostic samples. And the other point I'll make today is that we've shown that prolonged ex vivo culture of tumors may change functional biomarkers of drug response. So I'll first take a step back and get at the main question that motivates us, which is how do we pick the ideal therapy for patients with solid tumors that have otherwise failed standard of care therapy. Now, as Tony described, the conventional approach has been to extract DNA from a tumor and, based on the genomic sequence, identify the drug. The problem with this is that while sequencing has become certainly easier, faster, and cheaper, we struggle to bridge the gap between DNA sequence and the actual selection of the drug. Another approach that Tony described in detail, the functional precision medicine, aims to take patient material, expose it to multiple drugs, and then select the drug that kills the cell. And so while the results of functional precision medicine and functional drug studies are easier to interpret, these measurements are a little harder to perform. And so functional precision medicine has been around for a while, and there have been multiple technologies that have aimed that are there to bridge this measurement gap. The paradigm that we've been focused on in the Letai lab is personalizing therapy by performing early and rapid measurements of cell death. We think that one of the major benefits of these early measurements is that we don't experience a lot of spontaneous cell death and hopefully minimize changes that occur with prolonged ex vivo culture. An additional benefit of performing rapid measurements of cell death is that we don't have to expand the cells and we don't incur the cost and time to expand these cells. So Tony described a bit of how we actually do this. So as you heard, we use a technique called BH3 profiling. This measures whether a cell is primed for apoptosis, being on the verge of dying, or whether it's far from the apoptotic threshold, meaning that it's poorly primed for apoptosis. And this is the modification of this technique by Joan Montero, a very powerful modification that allows us to evaluate how a brief drug treatment increases the priming of tumor cells for apoptosis. And this is called dynamic BH3 profiling. And so this powerful measurement, in contrast to conventional measurements of cell death, which can sometimes take multiple days, this measurement takes as little as 24 hours. And so how does this work? I'll describe a high-throughput version of this protocol that we published a couple of years ago. But we effectively take resections of large tumor pieces from a patient or from a mouse model. We dissociate the tumor, plate them in 3D 4-well plates, and then we treat those cells with potential drugs. Now, after 24 hours, if most of the cells are still alive, we add an intermediate concentration of these synthetic pro-apoptotic peptides that Tony described to mitochondria. So if a drug in the well has no effect on the mitochondria, what we find is that the mitochondria remain intact after the addition of these peptides to the cells in mitochondria. However, if the drug primes the cell for apoptosis, the balance of these pro and anti-apoptotic proteins that Tony described changes, and then the peptide addition causes mitochondria to become permeabilized and release cytochrome C. Importantly, we can quantify the release of cytochrome C using automated imaging analysis, and we can rank the drugs according to the delta priming. And again, all of these details are in these papers, and if there's anything I want you to take away from this slide is that we can effectively rank the drugs that we think are going to prime the cell for apoptosis and the drugs that are going to be most effective in the patient as a single agent or combination, and these are the drugs with the highest delta priming. So an important question for us, having this technology at our disposal, is why do we make rapid measurements of cell death from patient-derived tumors, and why not just generate stable models of these tumors, either cells or avatars of these cancer, and then measure death or tumor shrinkage? So as a first step, we wanted to know whether there were differences between these these chemical sensitivities of fresh tumors and the chemical sensitivities of cell line models derived from those same fresh tumors. And so to test this, we performed dynamic BH3 profiling on a fresh tumor from a mouse, and then on a derived low passage cell line from that same tumor. Now, what you can see here is that we found differences in some of the drug-induced priming between the two. Notably, seeing a new vulnerability to the BCL-X-long inhibitor navitoclax in the low passage cell lines, and we also saw a relative loss of vulnerability to AZD2014, and this is an mTOR inhibitor in the established cell line. And so we could observe some similarities, but some changes. And importantly, when we actually put these drugs into mice in vivo, we saw that navitoclax failed to have an effect in vivo, but that the mTOR inhibitor appeared to work in vivo. And this to us indicates that established cell line models of cancer can be effective in certain situations, but they can also potentially provide misleading results. We've started to evaluate this in other systems, including here a colon cancer PDX model, where we see the emergence of BCL-X-long sensitivity in a week-long culture as displayed by the enhanced sensitivity to navitoclax and A133-1852. And we also see something similar with a primary GIST tumor from a patient, where we see again an enhancement of BCL-X-long sensitivity in a derived cell line that was grown out for a week, indicated again by the A133-1852 BCL-X-long inhibitor. So ultimately, these results indicate that prolonged culture can change chemical sensitivities and potentially provide misleading results when it's used as a drug response biomarker. So I'll skip ahead a bit, but having performed our high throughput dynamic BH3 profiling on large surgical resections of tumors, and just in general thinking about how to integrate our assay into clinical practice, we next wanted to determine if we could perform dynamic BH3 profiling and this high throughput dynamic BH3 profiling on diagnostic samples like hollow cord needle biopsies. And this is very important from a practical standpoint because doing this could potentially lower the barrier for us to sort of enter into the clinic and the barrier to uptake in the clinic. And so when we tried performing BH3 profiling on these needle biopsies, we failed. We failed a lot over and over again. And so we wondered, why did we fail? And it's fundamentally because what we did is we used our fixed time point assay, and in this assay, we actually need to identify this optimal peptide concentration. And if we don't, the assay is less informative and it generally performs poorly. And so unfortunately, to identify that optimal peptide concentration, we use a lot of cells, and we use a lot more cells than are typically found in a core needle biopsy from a solid tumor. So we struggled with this for a while, and ultimately to get around this, we realized that we instead should just add a high peptide concentration after drug treatment, and then instead of using a fixed cell assay, we should monitor drug and peptide-induced mitochondrial permeabilization with live cell microscopy. And this is just a schematic of how it works. We basically treat the cells with a drug and measure mitochondrial permeabilization using the potential dye TMRE, and doing this using continuous imaging. Now, for a drug that has no effect, once we add the peptides to the permeabilized cells, a relatively poorly primed cell will undergo mitochondrial outer membrane permeabilization at a later point. Conversely, for a drug that primes the cell for apoptosis in response to the peptide, the cell will undergo mitochondrial permeabilization fairly quickly and will lose its TMRE rapidly. So this is what some of our imaging data looks like. This is in pancreatic cancer cell lines. And so here, host is in blue, marking the nuclei, and TMRE is in green. And so you can see the peptide addition occurs at time 0. This time is in hours and minutes. And the cells which are TMRE positive at the beginning begin to lose it over time. And so we can observe that there's a rapid loss in the navitoclax treated wells relative to these DMSO treated wells. I'll play this again. And again, watch out to see that there is a faster loss in the navitoclax treated wells relative to the DMSO treated wells, indicating that this is a drug that's priming this tumor for apoptosis. So what's remarkable, this is perhaps a bit tangential, but even in cell lines, we noticed when we looked very closely, there was quite a bit of heterogeneity in even the drug-treated cell lines. And so if you look at some of these individual cells, you'll see some of them release TMRE early, indicating that they're quite primed for apoptosis, and some of them release TMRE late, meaning that after the drug treatment, they're still poorly primed for apoptosis. I'll play this one last time, but this zoomed in version, and just point out some examples. So these are two cells that are highly primed and release TMRE early, and you'll see some of the cells release TMRE, and then these are two cells that release TMRE at a much later point. So even after this drug treatment, there's a substantial amount of heterogeneity. We spent some time developing an analysis pipeline to capture this single cell information, where we can track single cells and evaluate their priming. This is just 10 randomly selected cells, but to get a sense of the population, what we could do is make this rainbow plot where individual cells are in the different rows. The time is on the x-axis, and the TMRE intensity is color-coded. And you can see that some cells lose TMRE early, and some lose it relatively late. And so to sort of quantify this, we can create a metric by looking at an area under the curve and evaluating the priming of these single cells. And again, there's more information in this paper if you want more details. So these are the rainbow plots of the video that I showed of the DMSO-treated cells and of the drug-treated cells. And you can again see that the navitoclax-treated cells lose TMRE quite quickly. So we can do this with live cell BH2 profiling, and we can do this with fewer cells. I'll skip over some of the technical validation and get to some of the meat here. But we next wanted to know whether this live cell measurement predicts in vivo response. And so before moving to patients, we wanted to test this in mice from Novartis. They gave us 7 different models with published in vivo responses to these different drugs. And we wanted to ask if these were 7 different patients, could we figure out the ideal patient population for a single drug? And so using DBP, we looked at unique pairs of models where there was progressive disease indicating a response to the drug, or indicating a lack of response to the drug, and unique pairs of models in drugs where there was stable disease or partial response. And we saw that there was a difference in the delta priming value of the live cell dynamic BH3 profiling between the responders and non-responders. It was great if we looked at the receiver operating characteristic of all of this. We found that this was a potentially sensitive and predictive marker of response. And just digging into this in a little bit more detail, drug by drug, we found that that delta priming using this live cell measurement could nicely predict responses to binimetinib, a MEK inhibitor. It did an okay job of predicting response to encorafenib, but did a pretty poor job of predicting response to CGM-097, which is an MDM2 inhibitor. And so this seems to work for some drugs and not for others. I think that's to be expected, and we can discuss more about potential reasons there. So I'll just wrap this up really quickly. Our ultimate goal was to do development where we could see if we could perform BH3 profiling using cord needle biopsies. We're still in the preclinical stage, and so we've used mouse models as a test case, and we've taken cord needle biopsies from the mouse. And we first wanted to ask the question of how many cores we need to recapitulate the delta priming in the primary tumor. And here we can see that we get similar results between 1 core and the bulk tumor. And again, we need to do more testing on this, and I suspect we need at least 2 cores, but we're seeing a decent correspondence between the cores and the bulk tumors. So just to summarize, drug response of patient-derived models can change drug responses with prolonged time ex vivo. This, I think, highlights the benefit of a rapid test. We can predict in vivo response using our live cell measurement. And importantly, we can now perform dynamic BH2 profiling when the starting material is a core needle biopsy, which we think is really important and in line with the type of material that we need from the clinic. So, I just want to quickly go over what we think is holding us back here and some potential challenges we have in the lab. So these are a few, including sample transport, robustness and reliability, which is problematic because we need to keep cells alive for 24 hours, and then just the flat-out demonstration that this live cell method actually works. And so I just want to quickly describe how we've tackled some of this robustness problem. And we've typically performed our assay at a 24-hour time point, and INESE joined us for a couple of months, and to sort of get after the question of what is the earliest time point where we could perform dynamic BH2 profiling. And we assumed for a lot of drugs, especially cytotoxic drugs, this would take 24 hours. And so they essentially asked, how long after you add a drug does does a signal take to get propagated to the mitochondria? And in SN38, which is a fairly important metabolite of an important drug, a DNA damage drug, INESE was finding that while we thought this drug would take more than 24 hours to read out a priming signal, that she could measure priming within 2 to 4 hours of drug addition. And we think this is really important because we do sometimes struggle to keep solid tumor cells alive just for 24 hours, and now we can shorten that down to a 2 to 4 hour time point. So a lot of people have contributed to this. I'd like to thank Tony, and then of course INESE, who did an excellent collaborator. And if you're from the region and want to do PhD profiling, I think she's, potentially the local expert now. So that's it.
Thank you. Thank you, Patrick, very much. And I'm inviting to the stage our birthday person. Happy birthday. Warm applause. Joan Montero, he's associate professor from University of Barcelona, from Spain.
Actually, I would like to start saying that it's an absolute honor to speak after Tony and Patrick, and thank you all for the birthday wishes. Oops. These are my disclosures. When we think of cancer treatment, probably the first thing that comes to our mind is a patient going to a hospital to receive a certain treatment. And obviously what we want when we give this treatment is to eliminate the tumor and cure the patient. This is the objective. The problem is that we all know that many patients relapse. So somehow some cancer cells are able to survive the therapy, and they may lead to relapse and disease progression. This is particularly evident in the case of melanoma, where this patient, for example, was diagnosed with a BRAF mutant melanoma, was treated with bemurafenib, and it first responded. But several months later, this same patient went back to the clinic with lesions that were more aggressive and no longer responsive to therapy. And I think that we can extrapolate that this happens in many, many types of cancer. What we and others have found is that anti-apoptotic proteins from the BCL2 family play a critical role in the cancer cell survival to treatment. In fact, what we and others have found is that in response to anti-cancer therapy, there's an accumulation of activator proteins such as BIM and BIT that Tony introduced. But the cells are able to adapt by using anti-apoptotic proteins. So basically, these anti-apoptotic proteins are able to capture these activators and avoid apoptotic cell death. If we are able to identify which anti-apoptotic protein is responsible for this adaptation, we can then add a second drug, a BH3 mimetic, in order to block this anti-apoptotic protein, release the activators that now will be free to engage back-and-back oligomerization and restore apoptotic cell death. In fact, BH3 mimetics, as it was introduced in the previous two talks, There are drugs that are currently explored in clinical trials, in hundreds of clinical trials. There's one that is approved, is ABT-199 or venetoclax, that many others are being developed to inhibit BCL-XL and MCL1. So it's a growing field, and we believe that in the next years, we're going to see more of these drugs being used in the clinic. One thing that you haven't heard yet about dynamic beach profiling is that apart from predicting the response to treatments, that's what Tony and Patrick presented, we can also use specific peptides mimicking the sensitizer proteins such as BAT, NOXA, or HRK in order to guide the effective use of BH3 mimetics. So basically, we can identify which antipoptotic protein the tumor is using to protect itself against cell death, and then use BH3 mimetics to block this antipoptotic adaptation. And while I was still a postdoc in Tony's lab, I started this collaboration with Rhys Hack. Rhys Hack is an oncologist at Dana-Farber that is studying melanoma. Melanoma is a type of cancer characterized by different mutations, but as said before, it's approximately 50% of patients present a common mutation in BRAF. And this led to the development and approval of BRAF and MEK inhibitors, such as dabrafenib and trametinib. What I tested back in the day was if these same treatments that are used in the clinic would also induce these antipoptotic adaptations. And to do it, I used a BRAF mutant cell line called A375, and I exposed to a BRAF inhibitor, dabrafenib, and a MEK inhibitor, selumetinib. As expected, because I was blocking the MAP kinase pathway, I observed an increase in priming with the BEAM peptide. And as you heard before, this means that these cells would undergo apoptosis. But what was more important, more shocking in this study, was that starting at 36 hours, precisely at 36 hours, these cells started to become MCL1 dependent. Or in other words, at 36 hours, these cells started to use this antipoptotic protein to protect themselves against this treatment. What we did next was to combine dabrafenib with an MCL1 inhibitor, and as you can see, dabrafenib alone only killed 20% of the cells, while the sequential combination of dabrafenib followed by this MCL1 inhibitor, AZD5991, led to the elimination of 90% of the cells. So we were able to multiply by 4 the effectiveness of this treatment. We also tested this combination in vivo using a xenograft model, and we observed that dabrafenib alone only delayed the tumor growth, while the sequential combination of dabrafenib followed by the MCL1 inhibitor was able to eliminate these tumors. And not only that, but we also detected a significant increase in survival in this match point treated with this combination. Again, demonstrating that this sequential combination could be actually really useful to treat melanoma. I next analyzed metastatic melanoma patient samples, and what I observed is that when these patients presented a BRAF mutation and were treated with BRAF, a MEK, or ERK inhibitors, there was an increase in priming and also an antipoptotic adaptation mediated by MCL1, demonstrating once again that this antipoptotic adaptation doesn't seem to be something happening only in vitro, but also in patient samples, and that in the future, we can exploit this antipoptotic adaptation pharmacologically using BH3 mimetics. But in this same study, I also analyzed several cell lines, lung cancer, GIST, and breast cancer, and in all cases, I observed similar antipoptotic adaptations. And this led us to think that these adaptations may be something more general and that may affect different types of cancer. And in fact, in my lab in Barcelona, we started to study pediatric cancers. Pediatric cancers are the leading cause of death during childhood. The therapy very often works, but even if it works, it induces devastating secondary effects on the kids that survive. And by definition, we saw it in some talks in the morning, there are a low number of mutations, so there is a clear lack of predictive biomarkers and new treatments. In my lab, we focused on 2 of the most common pediatric cancers, although they are considered rare diseases, rhabdomyosarcoma and pediatric BALL. Clara Alcon, a talented postdoc in my lab, studied rhabdomyosarcoma, and she tested the current therapies that are used to treat this disease. And what she found was that when she was performing a dynamic beach profiling analysis with vincristine, she observed antipoptotic adaptations mediated by BCL-XL and MCL1. And she identified particularly that vincristine synergized with the MCL1 inhibitor S63 And as you can see, there was a significant increase in cytotoxicity. And when she analyzed a PDX model of rhabdomyosarcoma, exposing these cells, the cells isolated from this PDX to vincristine, she also observed this MC1 adaptation. When we tested this sequential combination in vivo, we observed that this combination was the only one that was able to reduce these tumors, as opposed to vincristine that only delayed the tumor growth. Once again, we believe that these treatments could be useful, especially for relapsed patients. Albert Manzano, on the other hand, a talented PhD student that recently graduated, he focused on pediatric BALL. In this case, he studied new therapies, potential targeted therapies to be used especially for relapsed patients. And from all the targeted therapies that he identified as potential candidates to treat pediatric BALL, he found that trametinib and sunitinib were able to induce apoptosis, so were efficient when treating this disease. But he also observed that these treatments were inducing antipoptotic adaptations, and he specifically found that that trametinib synergized with MCL1 and sunitinib with venetoclax and with the MC1 inhibitor S63. Once again, demonstrating that these sequential combinations could be useful to treat relapsed BALL. He also analyzed a PDX model in collaboration with Klaus Debatin, and he observed that these same treatments, sunitinib and trametinib, were inducing this antipoptotic adaptation. And this let us think that these antipoptotic adaptations may be happening also in patients and could be pharmacologically exploited with BH mimetics. Since the publication of this work, Albert has been collaborating with pediatric oncologists in Spain in a network called RealNet. And he analyzed 6 relapse ALL patient samples. As you can see, he tested different compounds using dynamic BH profiling, and surprisingly, he found that venetoclax was a great drug candidate because it was inducing a clear increase in priming in all treatments. 3 of the patients that Albert detected that could benefit from venetoclax were finally treated with venetoclax. Basically, venetoclax was included in the therapeutic regime, and they all achieved a complete remission. Once again, demonstrating that this functional assay could actually help find better treatments, especially for relapsed patients. So basically, to summarize what we are doing in my lab, we are using dynamic BH profiling to identify potential sequential combinations of therapies. By therapies, I mean conventional chemotherapy and targeted therapies with BH mimetics in order to boost the effectiveness of the treatment. And here I'm going to switch gears a little bit. We heard from Tony and Patrick that there are different methodologies to perform dynamic BH profiling. We started many years ago with a fluorescence split reader. We next moved to a flow cytometry. That's the one that we use in the lab. And now we heard from Patrick, the high content microscope, which is the most sophisticated technique. All of them, they have pros and cons. But the biggest problem that they all present is that they require centralization. So you need specialized equipment and trained personnel to perform this analysis. And this is a handicap, especially if we think of implementing these techniques in the clinic. So what we are doing in my lab, what we started like some years ago, was to use microfluidics to miniaturize this technique. The idea is very simple. We believe that if we were able to miniaturize dynamic BH profiling, we could reduce the number of cells needed, especially thinking about non-invasively obtained biopsies, such as Patrick introduced. These microfluidic prototypes are hard to develop, but once you have the prototype ready, they are really easy to produce. You can basically print them in plastic, fabricate them in plastic, and they're very cheap. So this would also reduce the cost of this functional assay. Also, microfluidics has the advantage that could allow the dynamic BH profiling automatization, allowing also the in situ analysis. So at the end, what we want to do is to provide an accessible and user-friendly technology that could be implemented in hospitals worldwide to benefit patients. And this is the objective of this project. And this project was carried out by Albert Manzano. He worked with a microfluidic chip that provided a linear gradient. We need a linear gradient to perform those curve with a beam peptide. at least back in the day. Now with Patrick new technology, maybe we'll reconsider this. But basically, we obtained a microfluidic chip that was able to provide a linear gradient for especially thinking about using the beam peptide. As a proof of principle, Albert used two cell lines, GST1 that is sensitive to imatinib and GST1-670 that presents a secondary mutation and is resistant to imatinib. As a negative control, we use gefitinib. We first performed a flow cytometry dynamic behavior profiling, and we observed that, as expected, GST1 was the only one responding cell line, and basically we detected an increase in priming with imatinib and also cell death. But we couldn't detect it with the GST1-670 or when treated with gefitinib, as expected. Then Albert developed a system to perform this analysis, tracking mitochondrial permeabilization, that as Tony presented, means that the cells are undergoing apoptosis. And he used the fluorescent dye TMRE that tracks mitochondrial polarization and calcium AM to track the cancer cells. And what he found was when he exposed these cells to increasing concentrations of BIM was that, as expected, GST1, when exposed to imatinib, needed less BIM to undergo apoptosis. When he quantified the TMRE positive cells, as you can see, we could clearly detect a shift in the curve with increasing concentrations of BIM in the GST1 cells as opposed to the 670 or when treated with gefitinib. And as you can see, there was a clear increase in priming only in the GST1 treated with imatinib. We next tested this. We next used this same model using our microfluidic chip, and in this case, the beam gradient was produced automatically by the chip, and we observed something similar that in the imatinib-treated GST1 cells need less beam in order to undergo apoptosis. When we quantify the TMRE positive cells, we clearly saw this shift in the curve. That is what we need to detect the delta priming. And when we quantify it, once again, we observed that the only treatment that was inducing an increase in priming was GST1 when treated with imatinib. Again, demonstrating that this microfluidic chip could detect the delta priming in at least in cell lines. What we did next, in collaboration with the Sarcoma Translational Research Group at the Vall d'Hebron, was to analyze a GIST primary sample. This primary sample came from a patient that relapsed to imatinib, and we tested two new treatments, ripretinib, which is a second-generation KIT inhibitor, and dactolisib, a PI3 kinase mTOR inhibitor. As you can see, when we performed FACS-based dynamic behavior profiling, we didn't see any shift in the curve, so that means that there's no increase in priming. What we did next was to try to evaluate antipoptotic adaptations, as I explained before, and we found that especially dactolisib induced a clear BCL2 adaptation. So what we did next was to combine dactolisib with venetoclax, and as you can see, we could observe a clear increase in priming with this combination. As a proof of concept, we next tested these same treatments using the microfluidic device, and we observed that only dactolisib plus venetoclax was able to induce an increase on priming. And this demonstrates that this microfluidic chip could actually be used to identify effective treatments also in primary samples. To conclude, we believe that this microfluidic-based dynamic behavior profiling can identify effective treatments in cell lines and primary samples. The assay requires less than 24 hours in a small number of cells, around 30,000 per treatment. And we believe that especially this microfluidic approach, will allow the automatization and in situ analysis at a low cost. So at the end, what we would like is to develop a small instrument that could allow this analysis in situ to make it accessible for everyone. Finally, I would like to thank you all for your attention. Also, thank my mentor, Jeremy and Patrick, my collaborators, and especially Albert and Clara that did some of the work that I presented. So thank you so much, and I'll see you later at the panel. Thank you.
Thank you very much. Thank you. So we are moving, we started with the USA, then the Barcelona, so now we are moving a little bit closer to Baltics, and I invite to speak Professor Caroline Heckman. She's a research director of the Institute for Molecular Medicine in Finland.
Thank you, INESE, and thank you for the organizers for the invitation to come to Riga. It's actually my first time, even though I'm so close. But it's been a wonderful day, and I've learned quite a lot. But yeah, so I'm going to tell you a little bit about what we have been doing in Helsinki. And just to quickly say, these are my disclosures. They have nothing to do with what I'm presenting today. So basically, actually in 2010 is when we started our deep dive into functional precision medicine, or what we called individualized systems medicine. So we wanted to use a multi-tool or multi-omic approach to understanding drug resistance, mainly in cancer. And at FIM, we're a relatively new institute, and we were lucky to have quite a lot of nice tools in our institute. So basically, we had very good genomics tools, but we also had a very nice high-throughput screening lab as well. And at the same time, we have very good infrastructure for biobanking. And the head of hematology at the time, Professor Kimmo Porka, was very interested in setting up a nationwide hematology biobank with our help, and also with the Red Cross Blood Service. So really, our adventure started then, really trying to, at the same time, use the tools that we had at hand at FIM, but then also set up this national hematology biobank. And the idea of the biobank was that we would collect samples at different time points of the patient's disease and treatment journey, and store the samples so that the blast cells or the tumor cells would be viable after thawing. And this would be a centralized process where the university hospitals throughout Finland, there are 5, and then 24 regional hospitals would send samples to the biobank and be processed at the blood service and then stored at FIM. And then at the same time, if the patients desired and if there would be a clinical need, samples would also get sent to our institute, and we would do this individualized systems medicine study on them. And of course, the diseases we study in hematology are relatively rare, and in a country of 5.5 million people, there are not so many patients, so it did make sense to try to centralize this at the time, and also create this nationwide biobank and effort. And so we were really doing N=1 type of precision medicine studies at that time. But over the years, we've collected quite a lot of samples. So this started in 2010, and 13 years later, we have thousands of patients that have been recruited to the biobank. And we have analyzed hundreds of samples using our individualized systems medicine platform. And these studies have mainly focused on first one disease, which is acute myeloid leukemia, which was already introduced by the previous speakers. But at the time that we were starting this journey, the AML patients really had very little treatment choices, and the relapse rate was high. So basically, if you were a relatively young patient, so 65 or less, you could get intensive chemotherapy and maybe a bone marrow transplant, but there was really little else out there. But now, as we move forward in time, there are more drugs and more choices. But still, this is relatively few and targeting just a few actionable mutations in this disease. But then also, there are some new signaling pathway inhibitors as well. And you've heard a lot about the BCL2 inhibitors. So basically, around 2012, the first AML genome was sequenced. Over the time now, thousands of AML genomes have been sequenced, and really it has shown us that the disease is really heterogeneous, it's complex. There is no one driver mutation like CML, but there are many possibilities. And these different mutations are combined in different ways. And they also create then a challenge for treatment. In terms of applying the genomics for AML, what has been done is that there has been better risk stratification for this disease. So, for example, the European Leukemia Network now really just focuses on the genomics and also not only on point mutations, but also structural rearrangements as well. But what we have noticed is that we have an increasing number of patients that can now be stratified into what we call an adverse risk category. And their mutation profiles can be quite complex. But then if we go back to the older way of classifying AML patients, which is based on phenotype, this isn't really used that much anymore, but I'm going to show you that actually it can be useful. So just going back then to the tools that we have been using in what I call this ISM platform. So basically building a biobank of samples for diseases that are very rare, but then also applying our different omic technologies. So basically we started off doing whole exome sequencing and bulk RNA sequencing, but we have moved on to single cell sequencing techniques as well. And then you have also been introduced to functional assays, and for us this has really been focused on ex vivo drug sensitivity testing. In the beginning we were just using homogeneous cell viability assays, but we have moved on to single cell approaches using flow cytometry and also imaging-based technologies. So I'm going to tell you a couple of examples, and going back now to venetoclax and other targeted drugs. As we developed our technologies over the years and moved from homogeneous assays to more single-cell approaches, we were really able to start defining the cell types in these mixed patient samples that were being affected by these targeted drugs. So in a homogeneous assay, you just get one readout. You don't know what cell type is really affected, but in these types of diseases, you actually get a range of cell types. They can be very immature or primitive, or they can be more mature, and for AML, it's more of a monocytic, granulocytic type of differentiation. So just by flow, you can design antibody panels to look at the different cell types that are affected by these drugs. And just within a 48 to 72-hour incubation, you can see the impact of these targeted drugs on the different cell types that are in these mixed populations. So the BCL2 inhibitor venetoclax, for example, was very effective on immature blasts, but not so effective on more mature monocytic cells, while a MEK inhibitor like trametinib is actually much more effective on more mature monocytic cells, but relatively ineffective on immature blasts. So using drug panels where the drugs are plated in several concentrations, we could then get a dose response curve for each of those cell types. And if we then rank the efficacy of these drugs in the ex vivo assay based on a drug sensitivity score, we can see that, there is a very clear difference in these single patient populations. So basically, venetoclax was very effective on immature blasts, but not effective on monocytic cells, and then some of the other targeted drugs, trametinib or the JAK inhibitor, ruxolitinib, were much more effective on more mature cell types and not so effective on immature blasts. And if we just dig into the gene expression data for AML samples, and this is just using publicly available gene expression data for different AML subtypes. What we could see is that BCL2 itself is actually expressed more highly in the more stem-like cells, but the expression goes down as the cells mature. Instead, you see higher expression of BCL-XL and also MCL1. And this is also reflected in normal hematopoiesis as well, because the BCL2 family members are really important for normal blood cell homeostasis. So one story that came out, and this was really driven by a former PhD student in my group, Heikki Kusenmäki, was that just looking at publicly available data from the DepMap portal, where he was looking at the dependency of different AML cell lines on different BCL2 family members, there was a striking result in that erythrocytic leukemia cell lines were actually very dependent on BCL-XL. And so basically, these cell lines, which are all erythrocytic leukemia, were dependent on BCL-XL, but not so much on BCL2 or MCL1. And then looking at the Achilles RNAi data, he could also see that acute megakaryocytic leukemia cell lines were much more dependent on BCL-XL as well, rather than BCL2 and MCL1. So going back into the lab and then digging out all our erythroleukemia and megakaryocytic leukemia patient samples, he then ran a test to see if this sensitivity could be seen using BCL-XL or BCL2 inhibitors, and this was also apparent using these patient samples. So basically, using a specific BCL-XL inhibitor, the erythrocytic leukemia, megakaryocytic leukemia cell lines our patient samples were much more sensitive to BCL-XL inhibition compared to BCL2 inhibition. And then looking at the protein level, there was also looking at different erythroleukemia, megakaryocytic leukemia cell lines, also very high expression of BCL-XL compared to BCL2. So looking back into the genomic information of these patient samples, what we could see is that for the erythroleukemia patient samples, despite the presence of a P53 mutation, which is common in these two diseases, we still saw very good sensitivity to specific BCL-XL inhibition compared to specific BCL2 inhibition, and also very good sensitivity to both BCL2 and BCL-XL inhibition. And then this was also apparent when we applied single-cell sequencing to samples that were treated with a BCL-XL inhibitor, and both for erythroleukemia and megakaryocytic leukemia. So basically, you see a disappearance of the leukemic blasts when you treat the samples with a BCL-XL inhibitor, but really the blasts didn't respond well to venetoclax at all, but you did see actually a sensitivity to normal lymphocytes in those samples. And then going to a PDX model, this was a xenograft model, what we could see also is that specific inhibition of BCL-XL also resulted in inhibition of tumor growth. And then looking into drug combinations, actually what worked best was BCL-XL inhibitor in combination with a JAK inhibitor. And I think even more so, and what I'm not showing here is that actually pan-BCL2 BCL-XL inhibitor works quite well with a JAK inhibitor in erythroleukemia and megakaryocytic leukemia. So moving on then from what we have learned in the lab, a colleague of mine, Mika Contro, has worked with Heikki in developing the ex vivo testing to be implemented in a clinical study, and the results were recently published this year. So this was a phase 2 study. It was a 2-arm study looking at de novo AML, but then also relapsed refractory or secondary AML. And one of the motivations for doing this study was that, well, venetoclax is now approved for de novo AML if the patient is unfit for intensive chemotherapy, but it's not approved for relapse refractory AML, even though we know that there would be some efficacy. And so there was a motivation to set up this clinical trial so we could get venetoclax and use it for relapse refractory AML. So basically, this was a relatively small cohort of patients, but the patients all would undergo ex vivo testing, and then the ex vivo response would be compared to the clinical response. And what Heikki and Mika could see was that the assay was very predictive of sensitivity to venetoclax, and it was also very predictive of resistance as well. And the assay was just based on the flow cytometry ex vivo testing. They also used a conditioned media, which they found to be the most predictive of other media types that were tested. But basically, all patients that were refractory or had relapse disease also looked refractory in the ex vivo assay, while patients that underwent and met complete response, their samples appeared to be quite sensitive to the assay. So this is a way that we were trying to move the assay forward into clinical testing, and the assay itself is now being implemented not only in Helsinki, but also in Stockholm, Oslo, and Copenhagen. So the Nordic AML group is now working together to do a larger scale study. So this is now ongoing, and it's called the low-dose venetoclax study, so LD-VENEX, and basically they're seeing if we can reduce the amount of venetoclax for these patients. Finally, I just wanted to also mention that the results of our original individualized systems medicine project for AML were published last year, and there were 37 patients. These were all relapse refractory patients that were treated based on ex vivo testing results. And there was a 59% overall response rate, which is really striking for this group of patients who usually have an incredibly low response rate. And several patients were bridged to bone marrow transplant and are still surviving. So this is really showing that you can use this type of assay to start guiding treatment decisions. And then if there are people in the audience, especially students who are thinking, or other researchers who are thinking about implementing these type of assays, maybe not necessarily for clinical translation, but just looking at drug response and drug resistance, there are a lot of factors that need to be considered. We've been doing this now for years, and I think depending on the drug, you really have to think about the culture conditions and so on. And of course, there are different ways to perform the experiments and different instruments that can be used. And also the sample quality and quantity need to be considered. For our ISM project, that was a prospective study. We were using fresh samples, but now for our more focused subtype analyses, we've really been using our biobank samples, which have worked well. But these are factors that one needs to consider. And then finally, I think I can sort of say that the functional assays have worked for us, but we have really, implemented other technologies as well. And these have provided us ways to look for other molecular indicators of response. But all of these assays, of course, need to be validated in some way and tested in a clinical study. Which is being done now for the BH3 profiling assay now. But also, I just wanted to point out, because this is a meeting that has been organized by the Pediatric Oncology Group and acute erythro leukemia and acute megakaryocytic leukemia are both much more common in children rather than adults. I think what we're seeing in our hands is that the pan-BCL2 inhibitor, navitoclax, plus a JAK inhibitor are hopefully going to be quite effective for this subgroup. Navitoclax itself is moving forward in clinical study, but for myelofibrosis, and it's actually being combined with the JAK inhibitor ruxolitinib. So we'll probably see that that combination approved very soon. And all the work that I presented has been a very large effort by multiple researchers at FIM who are still there or who have moved on. So Ole Kalianiemi, whose work you'll hear about next, from Paivi, is now head of SciLife Lab, and then Christer Vanenberg, who used to lead our high-throughput core unit, is now at the University of Copenhagen. But multiple other people, Kimmo Porka and Satu Musyoki, and then also, of course, also the patients and our funders. So thank you for your time, and I'll pass the microphone on to our next speaker.
Thank you very much. Thank you. And I kindly ask you to bear with us for the last 10-15 minutes before the discussion. And I invite Associate Professor Paivi Ostling from Karolinska Institute to give us some highlights of the Sweden experience.
So thank you, Ines. I'm also very happy to be here. Riga is actually one of my favorite cities. I've been here many times and spent many lovely summer days in Jurmala. And I see I have 9 minutes and 45 seconds, so let's get started within this session. Next. It's not changing the slides. We need some technical assistance, some problems? I think. Yeah, so it's not changing slides when I press next.
Can I ask some technical people to help us?
Everyone's asleep. So we will wait a few seconds and they will switch to my presentation. So happy birthday, Joan.
Yeah.
All right, there we go. So I guess I have big figures in my presentation, maybe. All right, so I will give you some food for thought, some inspirational ideas on biomarkers in AML, including functional precision medicine, of course, being in this session, and then introduce, hopefully, if we have time, a functional assay in ovarian cancer, and then I have a few slides on going from infrastructure to implementation as well, but we'll see if we get there. Time is running. First of all, I would like to start to acknowledge the team that has done the work, specifically Tom and Brinton, who are our team leaders and run much of the research that we do in our group. I also like to remind us all that, like Tony said, precision medicine is a very wide concept. It would need to include, of course, the matching of patients to drugs. But we have many data types to be included in this and to be used for diagnosis, prognosis, and so on. And we will need to use AI to also get through and shift through and find the patterns in these data types. Working at a national infrastructure like the Science for Life laboratory in Sweden, we often have access to really vast technologies similarly to FIM in a way that you can use at your scientific problem. But that also comes then with questions that which technique really is the best one. And of course, you will always find a technology expert claiming that this is the method that you need to use for my cancer and to understand really what's behind. And of course, this is challenging not only to us researchers, but to clinical researchers as well, as to where do you really start and how can you really make sense out of this. And this is, of course, the challenge we all face, that initially these methods are expensive and create a lot of data. And how do we really get into this small line of clinical benefit? And how do we improve on, we are very good at collecting data, but how are we converting that into actual patient benefits? So this was, of course, also on our minds in initiating our systems precision medicine study on AML, which is done on diagnostic AML patients in this consortium that we initiated when moving from FIM to SciLife Lab. And you can see the partners of the consortium here. We have received samples, bone marrow samples, freshly and also frozen biobanked samples from both the Karolinska Hospital and from Uppsala, and done the simple sample preparation of extracting the mononuclear cells. Then we have used many of the technologies available at SciLife Lab and created around 90 million data points on around 300 patients as of now. And I'll walk you through some of the analysis that we've done. So the researcher running this project is Tom Erkers, my colleague, and we decided to try and define biomarkers for AML that are unbiased and that we are able to measure and quantitate. I will describe to you the data-driven hallmarks of AML. And we are currently finalizing this manuscript, so this will be unpublished data. So what are the data-driven hallmarks of AML, or what were the requirements that we wanted? Is that it was unbiased and data-driven, of course, and able to describe the complex biology of AML. And it should, of course, not only be genome, but also include the phenome and the functional parts of it. And most importantly, that you could actually quantitate and measure it, and it should be specific to this disease, and of course individualized. So this is the result of the analysis. So we have defined 11 data-driven hallmarks that you can see in this sunburst plot. We've given them arbitrary names that you can see on the sides, such as cytokine-associated signaling and differentiation cell states and so on. But you can also see that the many other methods that we've included, methylation, functional testing, mutations, proteins, and RNA, contribute to each of these hallmarks in a different way. So you can either have a positive or a negative contribution in your specific hallmark. And this is what the data behind one hallmark can look like. So if we zoom into hallmark number 2, you can clearly see this hallmark value in the blue dots increasing here, and it actually negatively correlates to venetoclax responses here. And you can see the underlying data on the transcriptional level, on the proteome level, DNA methylation, and on the mutations. And you could possibly conduct from this heat map that none of the single methods would be able to distinguish these patterns. Then, since we include drug sensitivity testing in the hallmark measurements, each of the hallmarks actually have a suggestion as to which category of drugs could possibly work if you have this type of a signature. And if we look even more closer on each patient, this is one example, or two patients, looking again at the hallmark number 2, where you can see a low value in hallmark number 2 would indicate that you are sensitive to venetoclax, whereas again a high value in hallmark number 2 would rather indicate that you are responsive to MEK inhibitors. This can also be seen, it looks really weak on the screen, but on a single cell level, so that you can look at the single cells in each of these patients and actually distinguish that they are different on this level. Of course, then running through the cohorts that we have, 118 in this particular study, you can create hallmarks for each patient in this way. But how does this help us translate, or what is the difference here? So, of course, we had a big debate in our group about calling them hallmarks to begin with, and it was Olli who decided that no, we're going to call them hallmarks. But it is important to distinguish that we are not measuring the Weinberg-Honahan hallmarks here. We are inspired by them. This is, of course, a very powerful concept within cancer research, but these are generic phenotypic properties across all cancers that are not as easy to measure and fairly hard to apply also in care. And this is not a personalized, quite the contrary, it's a general description of cancer. Whereas then the data-driven hallmarks are based on data. They are quantitative. It includes multi-omics integration on all different types of technologies, and it's tailored to the specific cancer type, and it's personalized for each patient. But also important to remember, we are not describing cancer subtypes here. So each patient has a hallmark, like a hallmark profile, including all of these on their own. So in a way, are we better placed to answer the clinical research in this way? So at least looking at how each of these methods contributed to the hallmarks, you can see that they contribute differently to each of the hallmarks. So if you zoom in again on hallmark number 2, you can see that the drug profiling, proteins, and RNA contribute to this signature a lot. So maybe, trying to get to my next slide again. There we go. So maybe the secret is that, yes, we do need to measure a lot. We need to do it disease specific. Yes, it will be expensive in the beginning, but if we have enough of these multimodal data on each patient and each a disease type, we can start defining composite biomarkers that are data-driven that we then actually can translate with cheaper and more simple assays such as PCR or such. Then moving on to the functional precision medicine in solid tumors, describing some of the assays and efforts we've done in this domain. Let's see waiting for the next slide. Here we go. So this is again my colleague Brinton Seashore Ludlow's work. So she's been active since we established the group in establishing many different type of high throughput methods to attack functional precision medicine. And importantly, we always use genomics to verify that our models are actually cancer models. And I'll briefly touch upon the 3D organotypic spheroids and the specific assay we have for those. All right, so this is drug testing to meet clinical timeframes. So how can we do that in solid tumors in the timelines that we need? So we just heard briefly on the 24-hour assay using BH3 mimetics. This is a different one. This also uses mitochondrial potential. So we measure with TMRM, but we also measure dead cells. And this is an image-based assay where you can see the DMSO controls at 2 and 72, and then the positive control, benzethonium chloride, which of course will wipe out all the cells. But this is an example on how it can look, and then we quantitate this in dose and actually calculate the dose response or drug sensitivity score that was developed at FIM based on the TMRM. And the assay is called the DETECT assay, and it's recently been accepted in precision oncology, but you can find the preprint already through the link in the slide. So in that paper, we describe 20 different patients. We've tested both tissue and ascites fluid. This is mostly high-grade serous ovarian cancer. And we've done both 2D and 3D and concluded actually that the 3D maintains the viability better than the 2D assays. But importantly, back to can we predict clinical response? So based on carboplatin measurements using this assay, you can actually predict if you have or separate the complete responders from the ones with progressive disease. This is also for the PFI, the progression-free interval time of either over 12 months or less than 12 months. And this is another example of a drug, a V1 inhibitor, that does the same. Interestingly, cisplatin does not work in this assay to predict outcome. But also then a number of hits, actually, the response in itself can be predictive. So to summarize what the DETECT assay can be used for, it works for both 2D and 3D, both tissues and ascites. It has short turnaround times, and you can either do it live or fixed. But importantly, you can actually then use it to design individualized combinations. So we usually save cells from the initial testing. We use the hits from the initial testing and then run combination testing, and in this way, within 10 days, we have specific individualized drug combinations for these patients. And we're now planning to implement this and start planning for a clinical test with these. So to summarize, since time is running up, I think many of the speakers before me said that relevant material, healthy controls, known drugs, and assay types are key determinants to make reliable results within FPM. Then I would also like to conclude that let's stop the technology fight. There's not going to be one method above all, but we rather need to focus on being creative about how we think about biomarkers and how that they are actually data-driven in a very broad sense. And that solid tumors is not impossible to do FPM on. It's of course more challenging, but it can be done, as been shown also by the previous speaker in this session. Then, being inspired by the research done partly in our groups and in other groups at Karolinska Institute, we also recognized soon that we need to gear our infrastructure to actually be able to meet this type of demand and to integrate data types across. And again, that genomics is not enough for functional precision medicine. Cancer or diseases are complex and need many different types of methods. And SciLife Lab, as an organization, then initiated a strategic team, and I have Åsa Johansson with me here in the audience today. She's my co-lead, a scientific lead for precision medicine at SciLife Lab, to define how we can restructure our infrastructure also to kind of meet the demands of precision medicine of tomorrow. And she will present some of our roadmap work more tomorrow, so I'll skip that out of interest of time. And I really thank you for staying here for the last session and the last speaker. Thank you.
Thank you. Thank you very much. And I invite all the speakers on the stage. We have like 10 to 15 minutes for the Q&As, the questions and answers. So while our speakers are coming up, it would be nice to see if some of the people in the audience have some questions. In this light, I don't see. Many are leaving. I understand there is a wine outside, so we can keep it short. Yeah, like majority. Like, can we turn on the lights? I know I'm going to be afraid to see how little people are. Oh, great. Thank you. So we do have the questions from the audience, please.
Hello, men and women. I have a question to Caroline Heckman. Nice to read your project, and I have questions about those 59%. What it mean?
Yes, thank you for the question. So that meant overall response rate, so 59% overall response rate. So this is not, say, long-term survival, but if there was a response to the drug that was identified, or the drug combination that was identified by ex vivo testing and then given to the patient, was there a decrease in the leukemic blast count, for example, and was it, yeah. So basically it's not progression-free survival or overall survival, but maybe a shorter definition of those. So basically there was a positive response.
Thank you.
All right.
There
Hopefully
is
I
a,
answered.
please, in front.
Great work, thank you. I was wondering, for the solid tumor assays, is there any way to build on your assays to predict immunotherapy response, or what cytotoxic would be synergistic with immunotherapy, like checkpoint inhibitors?
Yeah, so it's not, I haven't specifically worked with immunotherapies. There have been other folks in Tony's lab that have. It's potentially possible to build those in and to see how those work and whether we can predict response for those. But
Just like to say, I think sometimes we get a little too fancy in thinking what to combine with our immune therapies. Immune therapies don't kill via magic. They actually largely kill via programmed cell death pathways, such as exactly the sort of ones where all of us tend to measure. And we have found, and others have found, that almost anything that accentuates cell death signaling, any drug that accentuates cell death signaling, will actually facilitate the efficacy of immune therapy. So I think we can start there.
Thank you. More questions? Any hands up? No? I wanted to ask the question then from my side, and actually I was talking to some patient organization people outside before the session as well, and the question that the patients are interested in is like, how far are these functional tests to the actual use in the clinic? We can see like it's very variable depending on the test, so whoever wants to comment on this.
I can start. From my side, we, at least in Spain, we started to work with pediatric oncologists, and we are inside this RealNet network. So basically what we receive is patient samples from relapse patients, and we perform the NIH profiling, and we basically give them the information that we get, the delta priming for all the treatments, and they have more information to make clinical decisions.
But it's approved or not?
Sorry?
Like, do you need to be, like, this test to be approved?
Yeah, but if you have, you can, there are certain drugs that you can use as compassionate drug use. So that's an information that is especially valuable for relapse patients. And this is we're working now.
I can speak a little more about the United States. In the United States, there are several companies, actually, not just the United States, as well as Asia, and maybe one or two in Europe, that actually have assays that are ready to use. These are private companies, so it costs something to use their services. There's also clinical trials. I mean, obviously, that you've heard some of the speakers here at various centers in Europe. Tends to be more Northern Europe. Joan's an exception, but it tends to be more Northern Europe, and in America as well. There's open clinical trials for these sorts of things that you have to specifically search. I'd encourage you, visit our SFPM website, and we do have, you can look at some of the people who are doing these sorts of things, and that might aid your search.
And I mentioned this Nordic-wide study on venetoclax and azacitidine and AML. So yeah, we see that if we want to implement this assay, we really need to align our technologies, but we don't see that it would be possible to do it centralized, and really to try to streamline the assay as much as possible so that it could be implemented in a clinical diagnostic setting. So in this case, we're using flow as the instrument because it is used in clinical diagnostics anyway for these diseases. But yeah, the clinical trials do need to be done, so we're doing it in the Nordics, but then also our colleagues in Vienna, so Philip Stauber has been running trials in Vienna as well. But so, you know, trying to make it as easy as possible is also key, I think, to moving it to the clinic.
Paivi, you have some?
No, it's on. All right. No, I concur with the previous speakers in that easy assays are, of course, easier to implement, but I think also we could do a lot on the trial scenario in creating platform trials with standardized protocols where you can start a phase 1 as an academic researcher fairly easily once you have your preclinical data together. And I know of one cohort started by Sigrid Skånland actually for a drug combination in CLL included in the drug repurposing trial called IMPRESS in Norway, and I will be very excited to follow that journey again. So that's another way of getting into trials for these types of assays.
Any questions from the audience? I know you all want wine. And I praise you guys for being like so brave and so endurance that you stayed until the end. I wanted just to say thanks to our speakers, and I wanted to encourage everybody again. I know it sounds like advertisement, but SFPM homepage is really great source for seeing like what's new in functional precision medicine with different tests and with different approaches. And may I add that looking at some of the seminars, it's not always about the cancer. There are some other diseases sometimes included as well. So I would encourage you to at least see that website and find some useful information for you. So thank you very much. I think we can conclude our session and actually this day of the PMNET forum. And thank you again. Thank you.